High cholesterol is a significant problem around the world. According to World Health Organization, it contributes to about 2.6 million deaths each year. It is a huge problem in Canada too. According to Public Health Agency of Canada, nearly 2 in every 5 adults have high cholesterol levels. High cholesterol is a huge factor behind heart diseases and strokes as it can lead to a buildup of plaque in the arteries. So, to mitigate this problem and build a healthy and happy society, we present CholesterLock, a project that brings together a multidisciplinary team from iGEM Calgary that combines synthetic biology with engineering techniques to develop a novel solution to this issue.
So what solutions do current patients suffering from high cholesterol have to turn to? Lifestyle changes, such as improving diet and exercise, are necessary to reduce cholesterol levels. But pharmacological approaches are often needed for patients with a higher risk of cardiovascular disease. The most common and well-studied treatment for high cholesterol are statins, which are a class of drug that reduces cholesterol synthesis in the body. But statins are associated with numerous issues that prevent many patients from using them. Various adverse effects have been reported with statins, such as headaches, muscle weakness, and gastrointestinal problems. Among the more serious effects are drug-induced liver diseases, as well as increased blood sugar levels and risk of developing diabetes, making this treatment especially not viable for patients susceptible to liver and pancreatic disorders. So for patients with an intolerance to statins, their options become very limited. Ezetimibe is one of the most prescribed non-statin cholesterol medications. Ezetimibe targets the MPC1L1 protein to inhibit the absorption and transport of cholesterol in the body. But in addition to being less effective than statins, ezetimibe is associated with its own long list of adverse effects. Patients have reported muscle and abdominal pain, headaches, nausea, and it has also been linked to drug-induced liver diseases. What these current solutions highlight is the significant need for an alternative treatment for high cholesterol, especially for patients currently unable to take existing medications.
We propose to develop CholesterLock, a novel modified protein that can lower LDL cholesterol levels by inhibiting NPC1L1 similar to ezetimibe. NPC1L1 binds to and collects multiple cholesterol molecules before being activated and brought into the cell. CholesterLock will be designed with a cholesterol-modified N that will bind to NPC1L1 using a lock and key mechanism to block further collection of cholesterol, preventing it from moving into the cell. CholesterLock provides an effective alternative to the only available NPC1L1 inhibitor, ezetimibe. Unlike ezetimibe, CholesterLock has the potential to stay localized to the small intestines. Using synthetic biology tools, strategic breakdown signals can be incorporated to activate if absorbed or when entering higher pH conditions downstream of the small intestines. This could help limit the adverse effects common with other cholesterol-lowering drugs. CholesterLock, being a protein, has the potential to be expressed in probiotic bacteria and reduce costs associated with pharmaceutical manufacturing, making it a more accessible option for lowering cholesterol. To validate these benefits, we plan to talk to possible users and previous ezetimibe users about their opinions on probiotic-based alternatives to cholesterol medications and experiences with ezetimibe, as well as probiotics manufacturing companies to find an accurate price range. CholesterLock will be an easy-to-implement preventative measure to lower the risk of heart disease and can have additional benefits when combined with a lower cholesterol diet. In high-fat diets with lower cholesterol, inhibition of NPC1L1 was also shown to protect against insulin resistance, weight gain, and non-alcoholic fatty liver disease. The first steps in developing CholesterLock will require protein modeling software to optimize the design of the novel protein for the conditions of the small intestine and evaluate its interaction with our target protein. Our protein must be stable and have no secondary effects in the digestive system. We plan to discuss these factors with biosafety experts to get an informed sense of what to look out for. Next will be protein production. This will initially be done in E. coli along with proteins that can modify CholesterLock with cholesterol. NPC1L1 will also be required and will be produced using a cell-free expression system due to the difficulty of expressing membrane proteins in bacteria. To test the interaction between these proteins, they will first need to be purified using techniques like chromatography, and additional methods for dealing with the insolubility of NPC1L1 will need to be incorporated to prevent it from clumping in solution. The initial testing and development methods are designed to avoid the use of highly sensitive techniques with intestinal cell cultures or live mice to make better use of our resources and get more work done within the time constraints. The results of this project will lay the foundation for CholesterLock development and implementation in the future.
This project has been created with a focus on UN Sustainable Development Goals. For goal number 3 of good health and well-being, we recognize the negative impact that high cholesterol has on millions of people, and built our solution on reducing cholesterol consumption with the aim of promoting good health and well-being. Additionally, we looked further into goal number 10 of reducing inequalities by focusing on creating a product that has the potential to be affordable and accessible through a probiotic which involves synthetic biology-based production. In terms of production sustainability, we focused on goal number 12 of reasonable consumption and production, where this project aims to incorporate sustainable production through the use of synthetic biology rather than chemical manufacturing, as bacterial production uses less resources and is a more cost-effective process that can be implemented for large-scale production. Overall, to summarize, our solution in CholesterLock aims to lower LDL cholesterol levels by inhibiting NPC 101. This is an approach to treat high cholesterol and reduce the negative effects of LDL cholesterol consumption.